Well, thank you for coming back. I always appreciate you coming. This is lecture 10 in E340, 542. We're going to be talking today about the toggle circuit, gene regulatory network toggle circuit. In this case, we're going to see what happens to it when you apply forcing, which is known to be the case in its biological incarnations. And then we're going to look a little bit about how to simulate stochastic chemical reactions using the Gillespie algorithm, which is built into Tellurium. And then maybe, depending on how long it takes us to do the other things, I'll begin to talk a little bit about fitting and optimization, which will be the main topic for next week. As always, I need to say that the class is live streamed and recorded in edited form. So I'm going to review the little things that we did last week, reminders, and then we're going to do a set of exercises on the toggle. Now we'll jump back and see how to do Gillespie simulations. I'm not going to dig into the detailed mathematics of the Gillespie method. It's quite interesting. It's worth reading about if you're interested. And then we're going to do a couple of very simple exercises on Gillespie, and then do the toggle Gillespie algorithm, which is quite interesting in its results. And then perhaps, again, depending on how much time, there's this nice paper by Tyson, Chen, and Novak that has some interesting gene regulatory circuits in it. We might pick one of those and try to do the analysis we've been doing before on the hoof. And then, as I say, depending on how much time we have, I'll start talking about optimizers. So we could stop. I definitely want to get through exercise 10.10 today. I might cut out some of these earlier exercises if it looks like we're not going to get there. If we don't do the last couple of things, it's not. Those can be either put in a homework or delayed till next week. So in terms of coding, last week we talked about defining time-dependent functions using the colon equals syntax. Very simple change in our code, but it's useful to be able to do that. So you can then declare time variable parameters if you want. And that is because equal signs like K2 equals 1 are only evaluated when the antimony string is parsed. And so if you have cascades of K1 equals K2 times 2, K3 equals K2 times 3, K4 equals K3 times 2, and you change the value of K1, that won't actually update all of the values if you do it in Python. So the colon equals will do that for you. The colon equals has a problem, though, that you're not allowed to overwrite the value of K1 here from Python. So if I say K1 colon equals 0.2 plus sine of time, and then in my Python I say M2 dot K1 equals something, that will throw an error. I'm not allowed to overwrite it. So sometimes you have to think a little bit about how to use it, because you'd like to be able to do both. Okay, and then we spent most of last week talking about the toggle, cross-inhibitory toggle. We wrote down the base auto-activation case for A and B. We wrote a general form for inhibitory coupling with a Hill function where we had a variable strength of that inhibition. So I equals 1 means we have maximal inhibition. I equals 0 means we have no inhibition. It allows us to vary the strength of the cross-inhibitory coupling. And that gives us this set of equations that are a little bit elaborate looking, but are structurally pretty simple. We have that first auto-activation term, and then we have an and because we have multiplication, and then we have variable strength inhibition, And as always, when we're doing gene regulatory networks, we need to have a decay term, first order decay term. And this has the simple result that if we start with initial values of A and B, if A is big, initial is bigger than B initial, we go to a high value of A and a low value of B. If A initial is less than B initial, we go to a low value of A and a high value of B. If A and B start out equal, they stay equal, and we're on an unstable fixed point. And we did a little bit of bifurcation analysis. We wrote code that found the stable and unstable fixed points for these equations. And we looked at the bifurcations in B. That is the background rate of synthesis of A and B. And again, we've made everything symmetrical. It doesn't really have to be symmetrical between A and B, but it's easier to analyze if it is. And we showed that if the basal rate of transcription of B is very low, if it's too low, we get a steady low value of A and B no matter what we start with. There's a range of B for which we get either high or low value. The system behaves the way we expect it to. And then if we have too much basal transcription, the things turn on regardless of what the other one is doing. If we look in the context of the inhibitor, the strength of inhibition, as we would expect, if the strength of inhibition is weak, then we just have the A circuit by itself. In this case, for the parameter regime that we've picked, without inhibition, the circuit goes to A is high, always, regardless of the initial condition. If I reduce the value of B, I could get it to be low, regardless of initial condition, or bistable. And then as I increase the strength of the inhibitory coupling, I split into a high branch and a low branch of A. You'll notice something which I haven't really talked about in dynamical systems theory, bifurcation theory, but is classic, which is if I look at this line coming in of A star, you'll notice that the stable branch for small values of I becomes the unstable branch for larger values of I. And you'll notice that this line, this green line, is continuous. It's continuous and differentiable at the bifurcation point. So it exchanges stability from stable to unstable, but there's no discontinuity in the line. If I look here in the B bifurcation diagram, that line has a little bit of a bend in it, but nevertheless, when I cross the bifurcation, I don't see it jump. And that's typical of well-behaved dynamical systems. If the differentiability or continuity of the rate laws wasn't so well-behaved, then you could actually get discontinuities in the branches, but you don't get that here. And we talked about the classification of bifurcations. This is a classic pitchfork bifurcation. And here and for B, we have a classic, actually on the high side, we have a classic pitchfork bifurcation. On the low side, we actually have a discontinuity. And so that's actually inverse pitchfork bifurcation. All right. And then we did some phase portraits as we changed the value of the cross inhibition. And here the images were generated using XPP-aught. And I'm going to give you as an exercise regenerating these images with the null lines using our code instead. The code I shared with you and then the code we already had. And so, but let's just talk about what we're seeing here. If I look at the I equals 0.38 case, I see that I only have one stable fixed point where the green and the red line, they're pretty light, so they may be hard to see. On the zoom, the green and the red line cross. They only cross once. There's only one fixed point stable. Although I notice that the flow isn't just inward, there is some non-trivial flow direction there. At I equals 0.55, the red and the green null lines are essentially tangent at that fixed point. It's a little bit surprising. If I go up to I equals 0.57, and I should say, if these are correct, this bifurcation diagram that I'm seeing here for 0.38, 0.55 doesn't correspond to the one here. So there's got to be a parameter that's different between these two. Because here the bifurcation is happening at I equals 0.55, which is a little bigger. Things are pushed to the right a little bit. At this tangency, then I have some rather confusing things. If I look at my flow, it's upward here in the left-hand middle section. It's towards that old fixed point in the upward right-hand section. But I'm not seeing crossing of the null lines. And so they're the only fixed point is still the one in the middle. If I go to 0.57, now I actually see that my null lines cross 3 times. I have a null line crossing up here around 0.451, my old null line crossing at 0.5, and then the corresponding null line crossing at about 1.1, comma 0.5, a little bit less. So I said 0.5, it's actually 0.65. And my originally stable fixed point, it's a little hard to see. My initially stable fixed points become unstable, and those two null lines that have jumped, the null line crossings on the sides are now stable. Maybe not so easy to see that from the flow diagram. If I keep going up, then it becomes much clearer. I get these big butterfly wings, essentially, which are pointing to those two stable fixed points. If I push even higher to I equals 1, those stable fixed points get pushed all the way to the X and the Y axes. And so that's what's happening here. If I look at my blue line and I push I to be big enough, the fixed point goes essentially to 0, and the top one essentially goes to 1. It's pushing to the axis. All right. So my first exercise for you, and we can, as I say, this is one where once you have it, I'd like to have the code back because I can use it to regenerate some of these things, is to take, you should have code that will generate these vector plots already for the equations that we have. If you don't have that, let me know and I'll find it and share it with you again. But I'm pretty sure that I shared it before, or you may have your own version of it. And then I'd like you to use the null line code that I wrote to draw these red and green lines on the plot, so that we can essentially regenerate these images using antimony and tellurium instead of XPPR. So then for next year, I'll have a notebook where I can share and everybody can do it in antimony and tellurium and have these plots. Is that a clear exercise?
Yeah, of course. I mean, the code you gave me can draw both the flow and the null line.
Okay.
Yeah.
Do you have it implemented for these equations though? Because
No. I didn't type in the equations.
So what I'd ask you to do is take just a few minutes and implement the toggle equations at context, and then screen share it with me, and we can go over it together.
Okay, sure.
Now, capital K scales the range of the variables And so if you use a capital K of 0.5, the typical values of the variables are between 0 and 1. Now they can still go above 1. They could go to 2, 3. There's no limit on the top. But the interesting behavior will be in that range around 0 to 2, perhaps.
Okay.
If you used a capital K of 10, everything will be multiplied by 5. Essentially, everything scales linearly with K. It sets the scale range of the variables.
Okay.
Herbert likes to use K equals 10, capital K equals 10. It
Yeah.
does it basically just rescales the X and the Y axis. It doesn't actually change the bifurcation structure of the system if the K's are the same for the activator and inhibitor. If the K for the activator circuit were different from the K for the inhibitor circuit, then all bets are off and you can get more interesting, complicated behaviors. Okay, that looks great.
Yeah, the I is 0.338.
Great. And let's try a value of I that's a little bigger, like 0.75.
Okay. I didn't make these parameters as input. I just typed them as fixed numbers.
That's fine.
Let's see. On the ball.
Okay, great. And the other thing is that's the flow plot. If we have the vector plot as the one which is more like the one the screen we had, where you plot the vectors at each point rather than calculating the flow, would be good to put it on top of that one as well if we have it.
The vector. I don't have the code for the vectors.
Okay, so let's see. Let me find that. It's not in this one. Let's see. Open notebook. Yes. Okay, well, I don't have it. Okay, I have the general code for it, but I don't have it I don't have it for the with the toggle. But here's the general code. You paste that into a new cell and then replace the toggle the antimony with the toggle circuit, you should get it.
Where is it?
I put it in the chat.
Okay. Oh, here it is. Copy.
You won't need to re-import the right.
Oh, I need to.
Yeah, you'll need to replace that with whatever you used for the where you defined the antimony before.
Okay. I think wait, somewhere.
I have it here too, if you want it.
Oh, please, please paste it in the chat. I don't want to.
Oh, wait a sec. Let's hold on. Except I have, okay, here it is.
Okay. I just couldn't remember the location. There are so many files on my desktop.
Yeah, well, that's always the problem with finding things in Google Drive is always especially if they're shared with you. Finding them is a thing.
Um, Paste it.
And for that one, you're going to have to do a tiny bit of editing because you're going to have to make the if see if you look at if you don't have line numbers. Uh if you look down below where it says U and V equals J0, J2,
Oh yeah, right.
so you'll have to combine J0 and J1 and J2 and J3 for that to work. So at the end of the line for J0, you'll have to do a minus A. So if you
Oh,
if
okay.
it right there, I separated the synthesis and the decay. But for the purposes of that down there, yeah, you don't want those.
Okay.
You'll have to put that in those two lines.
So minus K1.
So
This is
yeah.
yeah,
And then you want to substitute whatever the other parameters were because I didn't. I had
K1.
I'd set K1 equal to 1 in that
Oh
one,
yeah, right.
but
So this should work. Oh, wait, I have to install it.
yeah, getting the vector plot to work is always a little tricky because getting the lengths of the vectors to come out right is a little bit fussy and getting the right number of them. Let's see what it did not like. Um,
And oh, there shouldn't and should not be there.
yeah.
Is that correct?
Let's see. Well,
No.
we've got the
Oh
range
yes, right.
is so you
Should
want
be 1.5.
yeah, try that.
This is too large. Wow.
Well, that looks. Let's see what happened there. Let me look at my code and see why. Oh,
100.
because okay, so if you scroll up,
Yeah.
you say so okay, you set X range, Y range to be 1.5, 1.5. Yeah, so we're doing too many points.
Yeah.
So 10 probably won't be enough, but that's fine to try.
20.
20 will be okay. And then we're gonna have to change it there. They should have had a variable so that we didn't have to
Yeah. Looks good.
yeah, that's getting pretty good. Okay, and so now the vectors are a little long, but we can always rescale that. But and now if we put the null lines on top of that, we should be able to see. So I'm going to do the same null line plot on top of it, null on top of that one too.
Then we can
You just called it call the null-line generator and then
That may work. No, it's overwriting the previous figure.
So you'll have to change that function call a little bit to do the null, just the null lines, not to do the yeah.
Yeah, I can.
Because it's nice to have both, because you see slightly different things when you have the continuous line for trade trajectories versus the vectors at each point.
Mm.
So it's always nice to be able to see both of those.
And the flow should not be there. Okay. Ow. Local variable.
Somehow numpy didn't get loaded.
Yeah, right. Let me delete this line. This line also. And run it from the beginning. Wait, what?
I'll let you debug. I'm not good. I'll get out of your hair and let you look at it for a minute.
Okay, now it works.
Okay, that looks great. Let's see. Is it great? Let's see. No, something is messed up.
Is it?
The scales aren't right. Look where your null lines cross.
But why?
Is that where your fixed point is? No. So the scaling of the x and y axes is different for the null line plot and the vector plot. And that's actually the problem I had in my old code, that the null lines were correct, but they weren't scaled to the same axes as the vector plot.
Hmm.
So it's interesting. Where the scaling worked for the flow plot, but not for the vector plot.
The vector is correct, right?
Vectors are right, and the shape of the null lines is right, but the
Yeah.
null line values are not scaled to the x and y axis properly. right?
Wow.
The crossing of the null lines should be at the fixed point, and it's not in that picture.
This is weird. There's nothing to configure here.
So somehow, let's see, lin space.
This is 0. This is 1.5. It's correct. Resolution is 4000.
Right. And when it's doing the plot, the axis labels are right. So let's look at the null line calculator, because I think the null line calculator somehow is scaling things differently. So where are you doing the okay, so that's the null line calculation, AX contour. That's not obvious how that can give you a different. Oh, this is generating the null lines, which let's look at the one, I think, let's look at the one that's generating the vectors. Okay, NP grid.
Oh, the I is different. Wait a minute.
Okay, well that would do it.
This is okay. Didn't expect. Some parameters must be different. K is 0.7, B is 0.5. Now B is 1. It should be 1. H is 4. I is 0.78. S does not exist. And K1 is 1. Yeah, this should work. Ow. Okay. S actually exists. Okay. Now it looks,
That looks perfect, yeah.
yeah.
Great. So now we have code that can generate both the vector and the flow plots, the null lines. It's a little kludgy because you have to retype things and you have to make sure the parameters. It would be nicer to have a cleaner code, so you only have to specify the parameters once, and but at least we could do it. So if we wanted to do this for a different system, we could do it pretty well. Great. Okay, good. Thank you. So let's let's uh, let's hold on to that because we may use that later.
Okay.
And save it so that if you can send me a copy back.
Yeah, of course.
Do you have a version that also plots the flows? No, it's you overwrote the one that was doing the flows, so you want to have both.
Two different figures.
Right. One with the vector plot and one with the flows, the way you had it first.
Oh, okay. Do you want it now or for the project? I can do this later because I have an idea. I can actually make it that you need you only need to type the equations once instead of typing it again here.
Right.
Yeah, I can make a script that translate this equation into this equation.
You may need the other one that we had, but that we started with again in the next couple of exercises, though. So I don't know if you want to save this as something as a different version and then do some undos to get back to where you started with.
Oh, yeah, of course. I will save this.
Yeah. So you have both versions of it.
How to share the, oh, here it is.
So you can just put my email address up in the top. Should find me. Be able to
I
share.
can send the link
Or
in
you
the
can
chat.
put the link in the chat.
Yeah, now you can have access to it.
Great. Okay. You want to take a minute to get the other one back, or do you want to, shall we keep going?
I can get it back.
Okay, I'll just give you a minute to do that.
Why
So
not?
it's nice to have both. So it gives you a lot of respect for Bard Ehrmantraut when he wrote the code his code to do this 30 some years ago. Now we have both on top of each other. So now we want to turn off the vector plot.
Just comment out this line. That's
Perfect.
good.
Okay, so now you can get both.
Mhm.
Perfect. All right. Thank you. So let me come back to let's come back to where we were. And I was able to get that share, so I've got a copy of your code. It's great. So we can now generate all of these nice pictures using our code, not having to use Bard's code. It was always a little bit embarrassing because Herbert's textbook, everything was in Antimony and Tellurium except those flow pictures, which he used a different package for. So now we can do everything ourselves. Right. And this was the same one which you already did. You actually did this one first. You did the flow one first and then the vector one. All right. So the next one, if you have it, where we did the bifurcation diagram here as we change the value of I. This one is interesting if you change the value of the Hill function. Now, as you'll remember, it's now possible to have the stoichiometry be variable. We're not actually using stoichiometry, so you could just override it. So you could just iterate, changing the value of h. So take the plot that you have to generate the bifurcation diagram, this bifurcation diagram. And this was initially we used h equals 4. Run it again with h equals 1. 1, 2, 3, 4, 5 maybe isn't so exciting, but you could do 1, 2, 3, 4, 8, 16, something like that, and see what happens. Because the reason I'm asking you to do this is that these Hill exponents are basically a representation of ignorance. We use Hill functions when we don't really know what the relationship is. We know something is low when it's low, it's high when it's high, there's some intermediate value. But we don't actually know the detailed shapes of those curves. We don't know those sigmoids very accurate. And in particular, usually we have to assume that the value of the Hill coefficient is pretty big, 4, 8, 16. And biologically, it's hard to justify high Hill coefficients for chemical reactions. And so you would like to know, clearly as I change the value of h, the bifurcation diagram will change, but you would like it to change in a well-behaved way. In other words, if the critical value of I moves a little bit, that's okay. If the high value of A and the low value of A move a little bit, that's okay. On the other hand, if the bifurcation disappears entirely, or the type of bifurcation changes entirely, then you worry that the model, the results of the model are very dependent on details that you don't really understand. And so it's a good idea when you do this kind of simulation to try running the simulation for different values of the Hill coefficient and making sure that you don't clobber your basic understanding. So do you have the code that generates the bifurcation diagram?
I have it.
Okay. So now if you can run that, ideally you would run it so that you put the curves on top of each other for different values of age, but it's okay if you run it and just have each plot have different value of age. So why don't you try that and see what happens as you change the values of age, especially if you have h equals 1, which would be Michaelis-Menten, or say h equals 2, 6, 20, and see what the curves do. And this is what's called the structural stability analysis. You're asking the question, when I change things that I'm not quite sure of in my model, that are a little bit arbitrary in my model, does the model give me results that are basically the same or not? And that's a very important thing to do when you're analyzing simulations. People often will build these complex simulations, they'll do parameter fitting, they'll get some result, and they'll say, I've got the result. But then you discover you change something a tiny bit, and you get a totally different result. And so you want to make sure that your results are robust to these kinds of uncertainties. Okay? Is that a clear exercise?
Yeah, yeah.
My explanation probably took longer than it'll take you to do the exercise.
No, I may spend some time to like find the code.
Okay. I added an extra flag to your call in your function gen null flow. So if I put a 0, it gives me the vector plot, and if I put a 1 in, it gives me the flow plot.
Okay.
So then I can do both with it. Don't have to use both. Okay. That works really well.
Okay, I got the part of the bifurcation diagram. I'm still trying to put them together into one figure.
Okay. If you get them in separate figures, that's okay too. It doesn't have to be all on the same. Did you get something that looked like this?
Uh, some of them.
Well, here I did 1, 2, 3, 4, 5, 6. So if you did different values, you might get slightly different results. But there are a couple of things to notice here. One of them is that as you make h bigger, let's start with h equals 1, which is the one all the way on the right. That's a classic pitchfork bifurcation. It says you get basically a quadratic splitting of the upper and lower branch at the bifurcation. As we increase the Hill exponent, the bifurcation happens for smaller values of coupling. So as the Hill exponent gets stronger, we get upper and lower branches for less cross-coupling. So effectively, the coupling is more effective when the Hill exponent is bigger. So that's important to know. Notice that the unstable branch doesn't change very much. For small values, the A star is a little bit different, but for large values, the unstable branch is almost the same, independent of the value of H. When we go to H equals 2, we see a discontinuous bifurcation. The values of the upper branch and the lower branch are immediately substantially different from the unstable fixed point. And that's also typical of higher order bifurcations. And also, as we make H bigger, the critical value gets smaller, but it will notice that the spacing between them gets less. They get closer together. And so if we used H equals 1 and H equals 6, they'd be very different. Although the general structure is the same, except for the fact that we have continuous bifurcation here and a discontinuous one here. On the other hand, if we used H equals 4 or H equals 6, well, the value of I at which the critical value, the bifurcation happens is a little bit different. But fundamentally, if we picked 4 or 6, it wouldn't make a big difference. And so what this is telling us is that even if we don't know all the details, the bifurcation is pretty robust to the details. One thing we could try is Herbert's form of this instead of my form. We could do the same analysis and ask how different are they. But remember that these kinds of network models that we're working with, it's not like particle physics or electrodynamics, where Maxwell's equations are Maxwell's equations. Here, everything we're doing is phenomenological. And so these equations are only very coarse approximations to the reality. And so if the details matter, then we have to worry that that we may get the wrong predictions. And so this analysis gives us a little bit of hope that we could be doing things right, at least that the results are meaningful. Okay, so the next thing I want to do is use that S that was in the equations that I put in the screen share. And at the moment, our A and our B are symmetrical. If I replace the letter A with the letter B and vice versa, nothing would change in the simulation. In reality, you don't have typically random choice of A versus B by cells. You typically have some external signal due to contact, due to a morphogen gradient, that biases the circuit in one direction or the other. And so we're going to add a source term, a signal, that favors A over B. And that's going to break the symmetry between A and B. In particular, in embryos, the cells that are on the outer surface of the embryo get different information from the cells that are further in. And that gives a bias to whether they're going to go high SOX, low oct, or high locked, low SOX. And so there are different ways we could include that activator. We're going to do the simplest possible thing, which is we're just going to say that S is a separate signal that comes in and increases the rate of production of A a little bit. And we're going to do that in the most naive way possible. It's basically going to increase the basal rate of production of A, and that means that we have B plus S, and everything else stays the same. There are plenty of other ways we could do this. We could make K1 here in that particular term a little less. We could reduce the degradation rate. But here, the easiest thing is to have S act as if it's increasing the value of B. And we don't have to change anything else. Okay. And that's already in the code, or you can add it in a trivial way. And now let's run the time series, go back to our old code, run the time series for this for initial values of A and B, different initial values of A and B, with different values of S. And before, what happened was that if A was bigger than B, you wound up with high A. And if B was bigger than A initially, you wound up with high B. If you have S turned on, then you expect that A will win, even if it's equal to or a little bit less than B initially. And so I'd like you to try that out and see whether that's what you get. Okay?
Okay.
And I don't want to talk too much while you're coding, but I'll say that one of the reasons I got so involved with this particular circuit was that a few years ago in the spring class that I teach, one of the students tried to replicate a paper that had been published by Ching Ni at Irvine and his students that was on this specific model, asking exactly this question. And we had a lot of trouble replicating their results. And so we spent a lot of time carefully going through it and redeveloping the code ourselves so that we know what was going on. But I guess that's about 10 years ago now, so it's not recent. But within the past 10 years, understanding this thing that we're doing here was publishable in PLOS computational biology. So actually this question is, it seems very simple, but it was actually a topic of current research investigation as recently as 10 years ago.
Mm-hmm. Okay, I got the result where even the A is starting with the value a little bit lower than B, it eventually gets higher.
Yeah, and depending on the exact value of S you use, that shift will be a little bit different. Here in my time series, a little bit like yours, I started with B, initial B always 0.5. And if I start with S is 0, if A is equal to B, they stay equal. If A is bigger than 0.5 initially, it goes high. If A is initially less than 0.5, it goes low. And here you see exactly that. If S is 0.4, I can start with A equal to 0.4, even 0.35, and I'll still wind up with that high solution. So it's not really very surprising, but it's nice to know that that works. So now, using the code that you just ran for that hill analysis, run the bifurcation plot for some different values of S. And go back to setting the hill exponent to 4, and now run the bifurcation plot for different values of S and see what happens. And as I say, it helps actually in this case. Normally we want to plot all three, a high branch, the low branch, and the middle and the unstable branch on the same plot. If you do that with this one, you can try that, but it gets a little bit confusing to see what happens. And so in this case, you may actually want to do high branch first, so big value of A, initial A value first, middle value of A first, second low value of A third, to get the high branch, low branch, middle branch, and then change the value of S. And it does something that's a little bit unexpected. At least it was unexpected for me. And I should say this is for bifurcation in the strength of the coupling, strength of I. So you're going to sweep the value of I. It could have been the bifurcation in B or something else. It's a bifurcation in I.
So is the curve uh shifting downwards when the S increases? It's in the chat. I think it's the lower branch. I only plotted the lower branch. Oh, wait, the x-axis should be I. It's a typo. The x-axis is I.
So this is what I got. Suppose it's possible I made a mistake. So let's take a look. I can say one thing already, which is if I look at the second one of these, where I see that the B curve has this mustache shape, that means the system wasn't run to convergence. And so the same thing here for the high branch. You see this curve, this little bit of a bend here near the bifurcation. And that indicates that the system hadn't converged properly. So that suggests a mistake on my part, at least in terms of the development of the simulation. It is going to depend on the parameter values, so we should make sure that you and I are using the same parameter values. And here I started with B0 equal to 0.6. But we could play with that a bit too. So here I am starting with A0 equal to 0.4, which is under 0.6. And if S is 0, I'm going to stay on the lower branch. So that makes sense. If I make S bigger, then I'm going to get A jumping up, which makes sense. If I increase the strength of the inhibition, then B has a stronger effect, and it's going to then tend to restore the A being low because B started out bigger, so it's going to be a race condition. B basically grows faster than A, and therefore B wins as you make the strength of the inhibition bigger. So why don't you see if that's what you're getting? It's possible I've got something wrong. Let's see if you can replicate these. Now, if I started with different initial values of A naught and B naught, it's possible I will get different results here. So in this case, there actually are other possible solutions beyond the ones that we have here. But the interest here is that if you turn S on and the inhibition is weak, the S dominates. If you make the inhibition strong, then the S has less of an effect. And so see if you can get, you don't have to be able to replicate all of those, but just see if you can get one of those to work for those parameter sets. One for that set of parameters. Pick something like S equals 0.5.
Okay. And what should the value of B be?
So in this case, I used an initial value of B of 0.6. But you can try changing that, seeing what you get.
The letter B.
Oh, little b. Let's see, what did I use? Good question. That's embarrassing. 0.5.
0.5, okay.
Let me double check that, but you're right. Should have been 0.5.
Hmm. I'm getting first half of the curve, but the curve is not coming back. Okay. Yeah, this is what I got. It's in the chat.
So in your case, let's see. So you're seeing, well, this is actually the upper branch, but you're
Oh,
not,
okay. Oh, yeah, right.
right. So this is, let me screen share this.
Okay.
So what you're seeing here is that upper branch in my A plot.
Oh, okay.
And you're right that as you're increasing the inhibition, you're not seeing it jump down again.
Mhm.
And that to me is interesting. And I'm wondering what happened to make it different. And I I was looking for my code from lecture 9 toggle bifurcation bias to check, and I'm not finding the code. So I'm going to have to look for that. So it's possible that I have a mistake,
Maybe
but
just the parameters are different.
or it's possible that the parameters are slightly different. Certainly for small values of I, these agree. You're seeing the values of the curves for A for small values of I are equal, but you're not seeing the jump down when the inhibition. And you're starting out with B0 equal to 0.6.
Yeah.
Right? What happens if you start out with B0 equal to 0.9, for example? Let's see what happens.
0.9. The capital B or the little b?
Capital B, initial value B.
Okay. It's the same figure.
Okay. Well, I will have to investigate that. I may have made a mistake. Simulation is an old simulation.
Oh.
So this was essentially my exploration of this. And again, it was trying to see when, let's see, maybe for these different values, I equals 0.4, I equals 0.8, S equals, okay, well, I'll, I will, because my analysis of this is the following, which is here, which is whichever one gets ahead first is going to win. And so my claim was that there's a competition of timescales. If F is big and I isn't too big, A grows faster than B. And then it gets to be bigger than B before you have enough coupling to commit irrevocably to one high, one low. And in that case, you get A high and B low, even though you started with A below B. On the other hand, if the inhibition is very big, then the B will win out against A. And as I say, that should depend on the initial value of A naught. So what happens if you take your A naught and you make it smaller? So run the same simulation that you just did, making B naught bigger and A naught smaller. You get the same thing.
I still get the same figure.
That's interesting. Because if I look here in this picture that am I screen sharing a slide?
No, you are displaying the figure.
Okay. So, well, then what I just said doesn't make any sense because I was trying to reference this figure.
Oh, okay.
So here, what I see is that here I is 0.4, S is 0.1, so it's weak. And here, if A is initially big, A goes big. If A is initially small, it goes small. B is initially bigger than A, just near A, goes high and then low. But what's interesting to me is this one where S is 0.6, and you see B shoots up here. It looks like B is going to win, but then A crosses the threshold, and even though B has gotten up to 0.7, 0.8, A is still able to outcompete it and knock B to a low value. And so that's the one where you see that the time scales matter. And again, I apologize, I'm not sure why we're getting different results. Why don't we try doing this now? Go back to our plots of the phase portraits, and now we can get better pictures than these, because these aren't so pretty. And let's make S non-zero.
Okay.
And what do you expect will happen? Well, you're going to get an asymmetry now. The fixed points of A and B are not going to be the same. And you're going to favor the fixed point of A. The region of attraction for the fixed point of A being high will be bigger than the one for B. So this is actually sort of an interesting one to play with. So now let's take the code that we had before, which had the S in it, and try this with S not equal to 0. So those are the plots we did at the very beginning of class today. Let's see what we get. Now, the way it's written, you'll have to change the value of S both for the null lines and for the plot, and you may have to change the scale of the X and Y axis, depending on the value of S used. So here, you didn't update the null lines to reflect the new value of S. And you'll have to change the X-axis scale to go to something like to 0 to 2. Your fixed point is off on has moved off to the outside of the field of view. Alternatively, you could just make S smaller. So here's an example. And again, I may have made a mistake, but here I had I equals 0.61, S was 0.24. And you'll notice that the null lines don't cross anymore here. And so the only fixed point now is this one for the large value of A. And so in this case, I wind up with a single fixed point. It's asymmetrical. Notice that if I look at the flow, it looks pretty similar between the two because I haven't labeled the direction of the flow. But you'll notice that on the right, on the top, if I start on the top left, before those would come down and then sweep up back to the left. Now they come down and they sweep to the right, and I only have a single fixed point. So see if you get something that looks like that. If you make S very large, then you push things far. Here we're just moving it a little bit. So in this case, no matter what my initial condition is, I go to the high A value. And that initial unstable fixed point in the middle has disappeared. Then you could decrease the value of S until they touch, until the green and the red lines touch, the null lines touch, and then you'll recover the old one. Still be asymmetrical, but you'll recover. Okay, let's take a look. Yeah, so that looks pretty much like what I have here, where even though if I'm far away from the fixed points, the flow looks very much unchanged. Because the null lines now that you hear your purple and orange null lines aren't crossing anymore, I only have a single fixed point. It's at x equals 1.5, y equals about 0.3. Why don't you see if you make s just a little bit smaller, try s equals 2.0, sorry, 0.20, and see if you can get find the place at which the purple and orange null lines just touch each other in the upper left. So here, yes, so here you've got for s equals 0.15, you've got the tangency of the two null lines. And so that's just the point where you lose that low A, high B branch. So, okay. So that's really what I wanted to do in terms of the analysis of the circuit. That's actually a pretty complete analysis of the toggle circuit. There are a few things that you could do in addition to that, but essentially we've done almost everything you could do with that. And now I'd like to move on in our last half hour or so, 40 minutes or so, and talk about stochastic modeling. So before I do that, I want to make sure the screen share is okay. You're seeing the slides again?
Yes, I can see the slides.
When I try to screen share your images, I sometimes forget to ship it back. So I'll make sure I do it. Okay. So there are many different biological systems are incredibly noisy. And they're noisy in a whole variety of ways, some of which we can model relatively straightforwardly and some of which we can't. One of the key ways that biological systems are noisy is that we're assuming that concentrations are continuous. But in fact, molecular reactions happen one molecule or molecular pair at a time. And those reactions are stochastic. Now, the rate laws that we are assuming make sense if the concentrations of the molecules are pretty big. But if we're talking about something like DNA, where we have one copy or two copies of the molecule, To assume that there is a density of DNA doesn't really make sense. It makes much more sense to think of it as an individual. And so there are at least two kinds of intrinsic noise, chemical reactions, one of which is one which we model all the time, which is decay. The decay of a molecule is usually exponential process. It's thermally activated process. And the probability of decay is usually constant during some time interval. There's no hysteresis. And so you get an exponential decrease of the concentration, or the decay rate is constant, and the decays are independent. To have molecules react to react, we have to get the two molecules, the substrates, to come together in some volume, and exactly how far and under what circumstances is a little bit mysterious how that works, how chemical reactions work. But in this case, the probability of reaction per time depends on the concentration of two species. Those probabilities are true whether we use a continuous approximation or not. However, as we know, when we do discrete versus continuous probabilities, you get Poisson rule effects, for example, that can do interesting things. You can also have fluctuations coming from outside. The concentrations aren't uniform in space, temperature changes stochastically in time, and so on. But those are both kinds of noise that you have to face. But the key one that we're going to care about here is that for many chemical species in biology, the concentrations are low. And so to assume that the chemical concentration is continuous and that the rate of reactions is fast enough that we can model the reactions as ordinary differential equations isn't so obvious. For a lot of signaling molecules, you might have 20 copies of the molecule in the cell. You can't assume that they're uniformly distributed. And so the whole idea of a concentration becomes a little bit questionable. And the idea that concentrations change continuously in time becomes questionable. And so the Gillespie algorithm essentially says, we're going to look at the concentrations of the two species. We're going to imagine that we pull from a distribution molecule of each species, and we say during a particular time interval, did a reaction occur or not, given the rate law? And that means that reactions are going to be stochastic and independent of each other. Now, the Gillespie algorithm actually calculates the time to the next reaction rather than how many reactions occurred in a fixed amount of time. You can have versions of the Gillespie algorithm that operate on fixed time steps, but the default Gillespie algorithm that Tellurium will provide us, the time will not be uniform time steps. You'll get variable time steps. And so that makes plotting and things like that a little bit more complicated. And so, as I say, there is an option to have it use fixed time steps if you prefer. If you're going to combine simulation replicas, you really need to do that. And if you're interested in the underlying science of the Gillespie algorithm, I recommend there's a good article in Wikipedia. You can go back to the original papers of Gillespie. And one problem with Gillespie algorithm is that it's slow. And another thing is that it's based on the numerical number of molecules. And the speed of the algorithm goes down as the number of molecules goes up. And so normally, so far, we've scaled concentrations to typically be between 0 and 1. We can't do that when we use the stochastic algorithms. A numerical value of 5 for the concentration is going to give us different results from a numerical value of 10 for the same rate law. But Tellurium allows us to change from modulo, the fact that we have to remember the scaling. Tellurium allows us to switch from the ODE solvers to the stochastic solvers with literally changing one word, which is instead of using the word simulate, you use the word Gillespie. And so if I type a model output equals model name dot Gillespie, start time, end time, and then if I want to force it to use a fixed time step, I type the time step. Notice this is now not the number of partitions I want, but the time step, and then I can select the output columns. Because we're counting molecules one at a time, we're going to need to increase our starting concentrations. If we start with 0.5 as a value, we have no molecules, so nothing will happen. And we also have to be careful because our rates have to be scaled so that they're less than 1. And so the scaling here is a little bit different, and chapter 6 in the textbook goes through all this in detail. But for tonight, I'd like to just run 2 simple examples to get us through the basics. And then I'd like to run the toggle with the Gillespie. So let's write a simple mass action decay law. A goes to B at a rate K1 times A. B goes to nothing at a rate K2 times B. Start with 1,000 of A and 100 of B. You can make K1 equal to 0.3, K2 equal to 0.1. And then we'll do result is r dot simulate, 0 to 30, 500 steps. Plot it. And then we'll reset it, and we'll set result is r dot Gillespie, 0 to 30, and we'll plot it and overlay it. Okay, why don't you try that one out? And see what happens. And if you'd rather use a different way of doing the plot, feel free. Okay, great. So what you got looked something like this. The stochastic version is a little bit noisy, but they basically follow the same curve. And so you would be, if you have 1,000 copies of A and 100 copies of B, for this parameter set, it would be legitimate to say that the continuum approximation, the ODE approximation, works pretty well. You may have a few percent error, but that's tolerable. So now let's go to fewer molecules. So try A naught equals 50, B naught equals 5. What happens? So we've reduced the concentrations by a factor of 20. Yeah, it's much more significant and it's a lot noisier. but the basic outline is still reasonable. Especially if I look at the A curve, it looks pretty much the same. They follow each other. Now try A naught equals 10, B equals 1. What happens? Yeah. So now, if I start with 10 and 1, well, the A curve, maybe I could get away with it, but the B curve looks nothing like the predicted value. Now, let's back off a little bit. 10 and 1 is a little too extreme. Let's go back to 20 and 2 and run it 10 times and overlay them. So what we want to do is we run 20 replicas, or 10 replicas, starting with A naught equals 20, B equals naught equals 2. Overlay them on top of each other. And what we're going to hope is that if we run this enough times, we have effectively more molecules, and therefore while the individual trajectories may not be exactly the OD predicted one, if we run enough trajectories and we average them, we should recover our OD results. That means the system is self-averaging. And fundamentally, we're not doing anything terribly bad by assuming the ODE.
And this might take a while because I'm modifying the code into the pyplot-lib version.
Okay. Yeah, you can wrap the code, just call the result. Well, the result you only have to call once. You can just wrap lines 25 and 26 with a reset. And then so all you really have to do is add a loop around line 25 and 26.
Oh, okay. So, but wouldn't it generate a new figure every time in the loop?
Well, if you use so the first time, well, what you want to do then is do a show equals false inside of here. And then after you've exited the loop, do t.plot array results show equals true again. And that'll do it. Or you could just do, I think, I don't remember, it's t show or something like that.
Okay.
But you can just call the t.plot array result with the show equals true outside of the loop and keep it show equals false inside the loop. So it'll plot one thing twice, but doesn't do any harm. That's the fastest way to do it. And remember, you're going to have to use that, put the r dot reset all inside of the loop. So you'd say after line 23, it would be just for a in range 0, 20 colon these two, and then repeat this line with this add of show equals false to line 26, and repeat line 26 below it.
Like this.
Let's see. Oops. put inside the loop, move the T plot array inside the loop.
Oh,
The
okay.
show equals false. So copy the last line, put it inside the loop with the show equals false, or just copy the T plot array line from above, paste it after result equals, and then leave it as it is. Be all right. There you go. Great. So if I look at this, I see that each time I run it, I get a slightly different trajectory. On the other hand, if I averaged it, the trajectories are going to average to the OD. But some things are going to be difficult. If I ask at what time did the concentration of B reach 5, there's going to be a huge set of fluctuations. The time at which things happen is extremely variable. So the overall trajectory will average, but things like getting the mean time to having something happen, what's called first passage times, don't average. All right. And so now the last thing I'd like to do, maybe we can talk a little bit, is go back to our toggle. And instead of running the toggle, and we'll just run the time series for the moment. We'll just run the time series. And substitute, instead of saying simulate, Gillespie. And the easiest thing to do is to start with the initial values of A and B of around 30, and to keep the equilibrium values around 30, make K1 and K1 about K1 over 30, whatever it was, smaller. And run that and see what happens. What does it do? And you can set S equal to 0 for this. You don't need the bias. And 30 is not unreasonable because it's not unusual to have 10, 20 copies of a transcription factor. So the actual number of A's and B's in the cell might be around 10, 20, 30. And you may have to move the capital K1 to being 15 instead of 0.5. Try it both ways.
Can I assign the accuracy, wait, the resolution of the simulation? I mean, the curve seems to be
So I'm sorry, you will have to, this should be capital K. It's lowercase k here, but this should be the capital K. K1, big K1. So let's see what you got here. Yeah, yeah, I'm sorry. So you're gonna have to start with capital, the initial values of A and B on the order of 10, and you'll have to make the Hill constant K, capital K, not 0.5, but to try 10 and see what happens. Because again, with Gillespie, if you have less than one molecule, nothing happens. I think you're seeing that. And you're going to have to increase the time range as well.
I think some of the parameters might be different. I got the oscillation, no, I got the noise, but I'm not getting the same figure as yours. Sorry, you are muted.
Sorry about that. I don't know. Sometimes it mutes when I'm not intending it to. Do you want to screen share it or put it in the, why don't we screen share it and take a look at what you got? See what.
Yeah, sure. Here it is. I was just playing around with the parameters.
That's fine. Let's take a look.
Yeah.
So I'm not seeing the plot.
Oh yeah, right. Here it is. Like this. It's
Oh,
loading.
so let's look at this. First, turn S to 0.
Yeah.
You don't want to bias. So make S 0.
Yes.
Okay. And then you want your K1 equal to 10. That's fine. Start with A equal to 110 and B equal to 10. So otherwise you can't see what's happening. No, not 110, just 10. Okay, let's try running that. See what you get. All right. Now something is not. Okay, let's go back. Can you move the figure so we can see the code? Okay. So you're going to have to make K2 smaller. You're going to have to divide K2 by 20.
Oh yeah.
Okay, try that. That's getting closer to it. All right, so now this is, let's see, we had H equals 4. Um, you have B, oh, okay, you have B and A both 10. That should be okay. A1 to the 2. What is different? Let's see. I equals 0.6, B equals 0.5. Yeah, that looks reasonable. Let's make the decay rate even a little smaller, make it 1/30. Take 2, yeah. There we go. Try that. Okay. So that's pretty close to what we have here, except that the time scale seems to be a bit different. So now let's take that Gillespie command and let's run it for 10,000 instead of 1,000. There we go. Now you got it. So the issue is, so for some reason, the time scale is different. So the K in front of everything, you have 0.7. Well, that's what I say here. So I don't know. I don't know why the time scales are showing differently, but in any case, that's all right. What you have is fine. So let's go back to your screen share for a second. And see the actual time series. So would you get this if you had ODEs? If you had an ordinary differential equation, would you expect to see this pattern of behavior? What happens with the ODEs?
If ODE is determined, the figure is determined.
Right. How is it different? How is the time series different?
It's random. It contains some random effect.
Yeah, but there's something very fundamentally different here from what you get when you run the ODE case.
Oh,
Why don't you just replace Gillespie with simulate, see what happens.
oh yeah. If we do simulate, there would be a stable state.
Right, exactly. So now try that. Yeah.
Oh wait, the scale is different. Not this one.
Right. Not sure why it doesn't seem to be displaying what you, it's not plotting what you expect on the ODE one. But okay. But yes, that's the key thing. So let's come back to this. Let me borrow the screen share back. So
Okay.
the key thing is, yes, if we use the ODEs, then either A wins and B is low, or B wins and A is low, and it stays that way for all time. Whereas here, A stays high, you'd say, well, here A1, 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 and then at time 600, it switched and B won. And the frequency of switching is very much dependent on the numerical value. If I make K1 equal to 10.5, over in this case for my simulation, again, I don't know quite why the time scale is different for yours and mine. In your simulation over time, my simulation over time scale 1400, I get one switch if I make K1 equal 10.5. If I make K1 equal 9, which is not a big change, I get 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 switches. In either case, I have a transcription factor that's supposed to be leading to stable differentiation of the cell. If I'm in high SOX, I'm one cell type. If I'm in high oct, I'm in the other cell type. And so what this is saying is that this circuit, in the presence of real biological noise, for the number of transcription factors that I really have, which is less than 30, will not give me a stable expression of the cell. The cell will switch type randomly back and forth. And the time scale over which it switches, well, we have to think about what the time scale means. But you find that the time scale over which it switches is not long. the time scale over which it switches is biologically meaningful. And so what this is telling us is that this circuit by itself cannot lead to stable differentiation of a cell type. If all we had were transcription factors with cross inhibition, then what we would find is cells would differentiate and then they would switch to the other type after a while and then switch back. Now, that kind of random switching back and forth does happen, but when it happens, we call it precancerous cells. And so if we had this kind of instability in our differentiation during development, we might be able to build simple organisms with 2 cell types, but we couldn't build a multicellular organism like a human being. Imagine if our neurons decided suddenly that they would switch into a different cell type and stop being neurons, or our muscles switched cell types. And if that happened on a time scale of 1 million years, we wouldn't care. Well, we might because there are a lot of cells in the body. But if it happens on a time scale of hours or days, it would be lethal. We couldn't build complex organisms. Now, you can play with the parameter values and maybe make the time for the switches a little bit longer. You could make the number of copies bigger, but biologically you're limited. The number of copies that you can have in a cell is limited, because a cell doesn't have that many, does not have the ability to have that many copies. And in addition, there's only one binding site for the transcription factor. So that number can't be big. And so this is what's called chatter. And in this case, no matter how many times we run the simulation, the results are going to be qualitatively as well as quantitatively different from the ODEs. And so this is a case where the ODE system lulls us into a false sense of security, thinking, oh, I understand how differentiation works. The circuit in this biology is known. The circuit actually does exist biologically. Transcription factors do behave this way. And they lead to either high A, low B, or high B, low A. Well, that seems reasonable. And over short times, that can work. But if we want to have stable change of cell type, this by itself cannot be sufficient. And in fact, cells have a whole variety of ways. When a gene is turned off, the gene will get methylated, which is a much more permanent way of turning it off. You basically go in and destroy the DNA, effectively. For things that are a little bit less permanent, the genes are wrapped, the DNA is wrapped on histones. Those histones can be acetylated, which promotes the transcription of the gene, or they can be methylated, which inhibits the transcription of the gene. And the methylation and acetylation of the histones is more stable. Still not perfectly stable, but it's more stable. And so the real biological systems have these backup mechanisms where, yes, A is high and B is low. And then there is some mechanism that says if A is being high for a long time, I'm going to add a lock onto it to make sure it stays high. B is low for a long time. I'm going to add a lock on it to make sure B stays low. And we almost never model those. And the main reason we don't model them is that we don't really understand how the temporary turning on or turning off of expression of a gene gets translated into these more permanent ways of turning it on and off. And so there's a big mystery still about how the circuits that we understand as gene regulatory networks actually lead to stable patterns of gene expression. It's not well understood. Now, something that you can do with this, and as I said, that was a homework assignment I used to give, but it wound up being a long time, is if you run these simulations for a long time and they're fairly slow, you can count what is the frequency, typical frequency, of switching between A high and B low. And you can plot that frequency of switching as a function of these parameters and ask the question, if I want to make the system more stable, what parameters are going to control that? Now, the easiest one is if I make the concentration of A, the high level, not 30, but 100 or 200 or 300, then the frequency of switching is very low. But that's a bit of a cheat. That's saying, well, if I were not in the stochastic regime, then I wouldn't have switching. So the question is, if I keep the range of A and B the same, what are the other parameters? Does H make a difference? Does B make a difference? Does I make a difference? Does K1 make a difference? K1 changes the overall time scale, so it does make some difference. But overall, what you'll find is that there isn't much you can do. You can't ever get rid of this chatter. And so I think that that's probably a good place to stop. Again, the key thing is that latches aren't stable. All right. So you're not going to have time now to try to do this kind of analysis for one of these other circuits. I'd ask you to look at this paper of Tyson, Chen, and Novak. And if I have time, I will try to give you a short assignment. But this paper, Tyson, Chen, and Novak, is nice because it gives you a bunch of different circuits, motifs. And now you have all of the tools you need. You can take a circuit, you know how to translate that into mathematics. You can take the mathematics, you can run the time series and see what the pattern of behaviors is. Then you can take that and you can do a bifurcation analysis on it. And then you can do the flow diagrams and the vector diagrams. You can plot the null lines. And so I'd ask you to go in and pick one, perhaps, of the circuits here, preferably not the toggle, which is the one we just did, and see whether you can replicate that. And that's a little homework assignment. But your main thing is going to be working on the P53 case. Okay? Anything else you want to talk about this evening?
Okay.
Any questions? Did
Um,
my explanation of the
no.
stochasticity make sense, or was that or do we want to talk more about that?
Oh, it is clear.
Okay. Because people I think don't appreciate that. In other words, I think a lot of biologists don't appreciate that.
But why? But the biology system is random.
That's right. And because the network analogy, the gene regulatory network diagrams look like circuit diagrams. And so we want them to behave like integrated circuits. We want them to be clean.
Oh,
so,
I see.
And if you run the ODEs or a Boolean network model, they're well behaved. But when you actually put in the noise that's really in the system, you discover they're not well behaved. And so there's always, this is a lesson that you can't assume that things that running at a lot of times will converge. And so that's why I did the first example where we did a bunch of cases. And while each case is a little bit different, if you average them, you get the result that you get for the ODs. In the case of chatter, you average it, you get nothing. You get a meaningless result. And so I think that that's something to keep in mind. And in physics, we tend not to do that. We tend to be in different regimes where we don't have to deal with that kind of problem so much. Now, maybe when you're dealing with quantum mechanical systems where the systems are intrinsically stochastic,
yes.
This kind of problem is something you run into, right? How do you get a well-defined result when you're in fact in some kind of superposition state? And so maybe for you, it's not. I mean, here there's no quantum uncertainty, but there is an inevitable uncertainty and noise in the system that we can't get rid of. And so we have to be pretty serious about understanding how do we have circuits that function even in the presence of large-scale noise. Okay, great. Well, thank you. I enjoyed talking to you today, and I hope that was fun. And let's try. I don't know. This week I'm supposed to be away Thursday and Friday, but maybe Friday. Send me an email. Maybe we can talk Friday about the project a little bit. I'll find a time to meet on Friday about the project if you have time.
Okay, Friday is fine.
Okay. Thursday I'm traveling, so I don't think I'll be able to do that.
Then Friday is,
In
yeah.
the end, I may not go. I may not go on the trip, in which case I'll be available Thursday too. But my current plan is I'll be away.
Mhm.
Let's try to find a time Friday when we can meet. Okay.
Okay,
Okay, great. Good to talk to you. Bye.
bye. See you.